Hi, I'm Christina Holder, and I'm a student at LeTourneau University. My research project is testing the reliability of two software called DNA Bind and DNA Binder to see how well they can identify DNA binding proteins in bacteriophages. DNA binding proteins are proteins that target and attach to DNA sequences to play roles in transcription regulation, host defense, DNA repair, and more. These proteins, like many others, are important to identify in genome annotation. For background, the idea of my project was born earlier this semester while I was doing functional annotation for the bacteriophage dynamo that I had isolated in the fall. As I was determining functions in a certain operon, which is pictured here, I noticed that most of the genes I had identified functions for were DNA binding proteins, and the rest were hypothetical proteins that had no clear function. So out of curiosity, I ran the hypothetical proteins in that region through these two programs called DNA Bind and DNA Binder. The result tables are shown here. The left is a prediction result from DNA bind, and the right is a prediction result from DNA binder. So the few labels that are next to these rows show the presumed functions of some of the genes in this area. I say presumed because the phage is not yet published. So the remaining rows that do not have a label next to them are all hypothetical proteins. And as you can see, many of those hypothetical proteins have a strong probability to be DNA binding. However, most of them did not have any strong alignments in NCBI BLAST or HH-PRED to DNA binding proteins, meaning I was not able to call a function for them. However, this did bring up the question of if I could trust these two software to make function calls in the future. So to test this, I chose 6 different clusters, which are listed here, with various hosts and life cycles. I then randomly selected 3 phages from each of those 6 clusters. I would load each phage in Famerator, a genome mapping program, and after I had the genome of each phage loaded, I would classify the genes in that phage as DNA binding or non-DNA binding. I did this by doing literature searches on the function of each of these genes by looking at various scientific papers, SEA-Phage forums, or other credible sources. I then ran all of the protein sequences through DNA Bind and DNA Binder. So now for a little bit about each software. DNA Bind is a program that uses the relative proportions of certain amino acids in a protein sequence in a linear formula to generate a probability that determines if a protein is DNA binding. This is a screenshot from the DNA Bind website. So as you can see, the software allows you to paste in a protein sequence here. I would also like to bring attention to the customizable false positive rate because that will be discussed later in the PowerPoint. DNA Binder uses an SVM model to predict a protein's DNA binding ability based on its amino acid composition. As you can see, DNA Binder also allows you to paste in a protein sequence. You may also notice that there are multiple data sets to choose from: main, realistic, and alternate. During this experiment, I worked with all 3 of these data sets. I would also like to point out that I worked with the amino acid composition setting, as the PSSM setting looks at evolutionary trends and can take hours to run. So after running a total of 270 sequences, 153 DNA binding, and 127 non-DNA binding, I was met with this data. This is very hard to analyze, so I decided to represent this data as sensitivity, specificity, positive predictive values, and negative predictive values, calculated through a software called MedCalc. So sensitivity is the percentage of DNA binding proteins identified out of the total number of DNA binding proteins run. Specificity is the percentage of non-DNA binding proteins correctly called out of all the non-DNA binding proteins run. So a higher sensitivity will mean that the software is identifying a higher percentage of DNA binding proteins, and a higher specificity means that the software is not calling many false positives. So we're looking for a high sensitivity and a high specificity. Positive predictive values shows the percentage of true positives out of all the DNA binding proteins the software call. The negative predictive values shows the percentage of true negatives out of all the non-DNA binding proteins called. So after calculating these statistical values through MedCalc, I represented them in these charts and graphs. So this is for DNA binder. And my first step was to decide which of the DNA binder data sets is the most reliable. So as you can see here, the DNA binder data set realistic, which is represented in green, has a high specificity, means that it called the least amount of false positives, but it also had a low sensitivity, meaning that did not identify many DNA binding proteins. And because of this, I don't think it would be very helpful in annotation. So this leaves the main and the alternate data sets. So as you can see, the main called more DNA binding proteins than the alternate data set, and it also called less false positives, meaning that the main data set is the most reliable for DNA binder. Next is DNA bind. So all the different rows you can see in the DNA bind table are the same software, but different false positive rates. So the default false positive rate that the software gives is 15%, which is relatively high. So I wanted to do some experimentation with lower false positive rates to try to maximize the software's specificity. So as you can see on this chart on the right, the false positive rates of 1, 5, and 7% all had a specificity of 100%, meaning that out of the 127 non-DNA binding proteins I ran, they did not call a single false positive. So that left me to debate which of these I thought would be most trustworthy. So as you can see, the false positive rate of 1% only identified 7% of the DNA binding proteins I ran, meaning it would likely not be very helpful. So that left the 5 and the 7%. And I would recommend that we simply use the 5% false positive rate, as it is better to err on the safe side and call less false positives in the future. So since these programs are not very commonly used, I don't think that either is completely reliable on its own. And because of this, I'm recommending that we run the DNA binder on the main data set in tandem with DNA bind run at a 5% false positive rate. This method would kind of mimic the protocol that we currently use for identifying membrane proteins, in which we would only call a function for the hypothetical proteins if they were agreed on by both software. This would ensure that we're not calling many false functions during annotation. So to show an example of this method in action and to kind of come full circle from the start of the presentation, I decided to use this procedure to try to identify DNA binding proteins from the hypothetical proteins previously discussed in Dynamo. So these two tables are the same ones that were shown at the start of the presentation, which show the prediction results of DNA bind run at a 5% false positive rate and DNA binder run on the main data set. So as you can see, there are blue arrows that point to proteins that are called to be DNA binding by both software. So as you can see, out of the just 11 hypothetical proteins in this region, using the software would be able to provide a function for 4 of those 11 proteins. This could be very helpful in annotation. So in the end, both DNA bind and DNA binder had fairly low sensitivities, all below 65%. Sensitivity and specificity for each software was either dataset dependent for DNA binder or false positive rate dependent for DNA bind. Both software had fairly high sensitivities and fairly high positive predictive values, which are all shown here. So overall, using the DNA binder run on the main data set and DNA bind run with a false positive rate of 5% would likely be very helpful and fairly reliable for identifying functions for some of the hypothetical proteins in bacteriophage annotation. So in future research, I would like to expand my data set to encompass new clusters and new gene functions to get more broad data. I would also like to do wet lab experimentation to test the DNA binding ability of the hypothetical proteins called to be DNA binding by both software to get more concrete data on the accuracy of these programs. I'm also thinking about looking into the reliability of other protein prediction softwares such as DNA Star and AlphaFold. I also think it could be helpful to look into creating a DNA binding protein prediction software specifically for bacteriophages that could be more accurate than DNA bind and DNA binder. I would like to thank the SEA-Phages program and Howard Hughes Medical Institute for hosting this conference. I would also like to thank Dr. Hatfull and the University of Pittsburgh for helping with phage isolation and sequencing projects. I really appreciate the LeTourneau University Department of Biology and Kinesiology for funding this project. And I'd like to give a special thanks to my professor, Dr. Ballrain, and the TAs in my class, Christina Spencer and Angela Salazar, who helped me and encouraged me throughout my project. Thank you for listening.